Hello, everybody, and welcome to Sick AP Bio Review 4.1 to 4.4. Here we are dealing with cell communication. And as I graded the FRQs, I realized a lot of people messed up a couple of key things. So we're going to focus on them right now. 4.1 is cell communication. Here it's all about generating and transmitting and receiving, responding to chemical signals. So we're going to describe the ways that a cell can communicate with one another. Now, cell communication deals with typically a cell releasing some sort of material, whether it is a hormone or some other type of protein or some sort of steroid molecule. That will be known as a ligand. It will leave that cell. It may act upon that same cell, or it may act on another cell touching it, or another cell near it, or another cell far away, all things, through something like the bloodstream. So basically, these cells can possibly communicate directly, possibly direct contact, or over a large distance. So let's take a look. Juxtacrine signaling is when cells are in physical contact, aka they are touching. In the world of plants, that juxtacrine signaling is going to be done through plasmodesmata. That is a way that plant cells can communicate with each other when they're physically touching. In animal cells, they're going to see something called gap junctions. And these guys are similar to plasmodesmata, except there's no cell wall to deal with. And these are channels between cells that are actually touching each other or next to each other. Let's see what we got. If you notice in both of these examples, this, let's say, is cell number 1. This is cell number 2. They are physically in contact with each other. And there's that channel right there. That's your plasmodesmata. In the world of animal cells, this would be one cell right there. This would be the second cell right there. And there is the gap junction channel. And they can either be open or closed. So they're either going to let materials in or they're not. Now, in the case of cancer and whether cells grow or not. And again, these here are super detailed. You're not going to need to know any of these specific names. And if you do, they'll give you everything you need in the question. But a CAM, or a cell adhesion molecule, is how cells know they are actually in contact with each other. So, for example, you have your skin cells and they grow in a pattern and everything's okay with them. When you get cut, these cells are no longer in contact. These cell adhesion molecules, these kind of connectors, which you see here, they've been severed through being cut or whatever. And in that case, your body says, all right, let's make some more cells and fill that gap up, which may cause a scar depending on how bad the cut is. If cells are touching each other, these CAMs are basically saying, all right, we're cool. Now, sometimes if the cells are touching each other and these CAMs are there's a signal malfunction, that could lead to cancer. Again, it'll be in the question with all of these specifics if they want you to know them. But keep in mind, a CAM is simply just two kind of extensions that let cells know they are in contact with each other. Now, in an immune response, we did not talk about the immune system like as a system, but what happens is there are these immune system cells. And what will happen is they're going to sense a problem. Maybe it's an antigen, a foreign antigen. And what they're going to do is they are going to that antigen is going to be picked up by a receptor in this special cell, which is then going to become known as an antigen presenting cell. And what will happen, you're going to see these CD4 and CD8, these T cells. These things are going to be created in an immune response. So these macrophages, which are these big cells that ingest foreign antigens and digest them, they take the pieces and they present them on their surface, known as antigen antigen-presenting cells. And what will happen is they will produce these T cells, and that will help fight future infections. Now, in the event that that antigen is a foreign organ in your body, that could be pretty detrimental to you. So hopefully it doesn't happen. But in the event of an antigen in the air or a pathogen, this is a way to help protect you. Paracrine signaling is type 2. Now, these guys are extremely close to each other, but they do not touch. So close, no touch. And in this case, a great example would be a nerve cell or a nerve to a muscle cell. So for example, if this is a nerve cell and this is a nerve cell, that's fine. That's going to be how your cells communicate with each other. And there's a space there called the synaptic cleft, or some of you may just know it as the synapse. And these little green dots, I guess they are known as neurotransmitters. So you will see here that one cell is the receiver with the receptor, and one cell is sending a signal. This will be known as the presynaptic cell. This will be known as the postsynaptic cell, and that's how information gets from one cell to another. Very, very close, but not touching. So with that, when you were looking to explain, you needed to say something like they were close without touching, and an example would have been neurotransmitters. That would have been a perfect example. Other examples of paracrine happen in bacteria, and it's known as quorum sensing. And what's kind of cool is the bacteria cell will release these chemical signals. Now, what will happen is if there's not a lot of bacteria around, these signals kind of just go off into nothingness and nothing happens. But if there's a large enough population, if there is enough of these bacteria all producing these chemicals, there's a reaction. And in a bacteria known as Vibrio fischeri, that response is a bioluminescence, where the bacteria light up, but they have to be in extremely high concentration. So here is I don't know why I put these out of order. I'm sorry about that. So this would be a neuromuscular junction. So this would also be paracrine. This would be where a nerve cell contacts a muscle cell. Well, it doesn't contact, but it's very close. It communicates with a muscle cell. I don't know why I put that in the wrong order, so I apologize on that. Endocrine signaling, this here is long distance signaling. So you needed to say in your answer, if you talked about endocrine, something like a hormone and something about the bloodstream. So if you said something about a blood vessel, if you said something about bloodstream, something about blood, that would be perfect. Taking a signal from one part of the body to another part of the body, a signaling molecule being released, acting on a target cell somewhere far away or over a long distance, something along the lines of that. If you said that, then you got your points. So that is the cell to cell, one cell to a different cell communication. Hormones are great examples, and we're going to talk more about feedback coming up in feedback loops. But here, the hormones, something like insulin or something like growth hormone, they will be released by one gland and they will act somewhere else. Other ones you may see testosterone, estrogen, thyroxine. These are all hormones released into the bloodstream. Endocrine. Nice little review here. Autocrine goes to yourself. And this here is, I don't know why they don't have it there. This is your juxtacrine. This here is your paracrine. This here is your endocrine. And again, a nice little review. Has all of the things that you need. Talking about signal transduction now in 4.2. Now here a lot of people kind of missed one little thing here. So what are the components of a signal transduction pathway? Well, they all have to have some sort of reception. Now that reception can happen inside of a cell or outside or on the cell membrane. And it's going to be the reception of a ligand. Well, that ligand could be a protein or it could be a lipid. Now, this is a very big deal because there's going to be two very different places for reception. The protein-based ligands need a receptor because they are unable to enter the plasma membrane. This may be because they are polar. And we know that polar things do not enter the cell. Now, these pathways may include some protein modifications and some phosphorylations, which we will see. Now, a lipid, which we'll see coming up in a little bit, can get right through the cell membrane. So if that's your cell membrane right there, and this is your receptor kind of in the cell membrane, if this is a protein, it simply will stop right there. If this is a lipid base, what's kind of cool is, there's your lipid base, your receptor is actually inside of the cell, so that can pass right through the cell membrane because it's non-polar. Always remember, protein's polar, usually large, but the size doesn't really matter here, and they cannot get through the plasma membrane, but the lipids can, or steroid-based. So if you look here, I added this. You don't have this, so in the video you'll see there. The ligand or hormone or signaling molecule, whatever you're talking about here, if the reception is in the plasma membrane, you know it is a protein or a hormone. Here, the same thing. This signaling molecule never enters the cell. There is the receptor built into the cell membrane, so you know that these are proteins. Now, in both scenarios, that's going to trigger this transduction or this cascade that will happen inside of the cell. But the molecule itself does not get in. That is extremely important. And in both cases, everybody got this right. There is some sort of cellular response. So what do we got here? In the first one, the pathway is off for whatever the signaling pathway is. And once again, it doesn't matter. Just read the picture. If there's no ligand present, there's no response. No big deal. Everything's off. But in some cases, watch what happens. Here, there's your ligand. So you know it's a protein because there's your receptor on the cell surface. One change is going to turn on another change, which is now going to signal this cascade of things. Notice there has been no cell response yet. Finally, there's your cellular response. So you have your reception happens here. This one and this one and this one are your transduction. And then finally, you have your response. So this is a nice little kind of summary of what's going on. Well, what are some of the things that could happen? Well, you could have your ligand bind, which is right there. And you know this is a protein because it's going to bind right into that receptor, which there's, again, much more to the story here. But there may be something called the G protein, which may kind of slide on down, which then may activate this other protein that's sitting here in the plasma membrane, which is going to take ATP and turn it into cyclic AMP. And what you're going to notice here is this is going to happen a lot over and over again. There's going to be a protein kinase. You're going to be turning on steps. Also along the way, this is going to amplify. And that is important. More and more and more until finally, there's your cellular response. So there's your reception, there's your transduction, and there's your response. So after the ligand binds, there could be something called the G protein. Once again, that'll head to the adenylyl cyclase, taking ATP, turning it into CAMP or cyclic AMP, which could activate kinases and ultimately a cellular response. Another thing that could happen is something known as a phosphorylation cascade, where you just keep on, as you see here, as you're moving through, you're just phosphorylating along the way. Here it is again, causing a response. Now, once again, you'll not have to memorize all this stuff, but you will have to understand it because they may give it to you in a question and they may say, well, what happens if this step is blocked? Well, if that step is blocked, the transduction doesn't happen. And if the transduction doesn't happen, the cellular response doesn't happen. So those are the things that you're looking for in these questions. All these fancy words, just read them and watch as the story progresses. Now here's that lipid or steroid base I was telling you about. Notice it just passes right on through the plasma membrane, and the reception actually happens in the cytoplasm. Then the signal gets taken into the nucleus, where there will be a cellular response, and that cellular response may be to create some mRNA. You're tricking the DNA, which is then going to build some new proteins, which could, again, maybe make muscle cells huge and anabolic steroids, things like that. So in the world of steroids, these are non-polar, and they can enter the cell membrane. Don't get hung up in size. It's all about polarity. Once in there, they can cause a cellular response. So again, look at the picture carefully. Notice the difference. So here we have, perfect, non-steroid hormone, otherwise known as a protein hormone. Notice it does not get in. In the steroid hormone, it does. So this picture is really cool. And again, in this case, the cellular response is to make a new protein, which is fine. In this case, the cellular response seems to be taking this complicated looking molecule and turning it into this linear molecule. And again, it doesn't matter what it is. Just read it, look at it carefully, and follow the story. Other, again, interesting things that could happen. The ligand could bind. It could set off this series of events which are going to open channels. So you may have a calcium channel open, which is going to let calcium out, which could then bind, which could cause a cellular response. Remember, don't memorize it. Follow the story. So once a ligand binds, for example, some channels will open. Just follow the story. How about some signal transduction? Let's put it all together. So, describe the different types of responses that could happen. Let's see. Quorum sensing. We talked about this. This is where the bacteria, as we saw it in 4.1, where the bacteria release chemicals into their surrounding environment. If there is enough population density, you'll notice here, it's going to be hard to see the red. Oh, no, there it goes. You will notice there will be a glow. And that is, once again, the Vibrio fischeri. Should have capitalized that V. Sorry about that. Other examples of things that could happen are the ripening of fruit. So ethylene gas. Now what will happen on a real tomato plant is this one will release a little bit of ethylene, which will then ripen that one, and so on. But what if you had a whole bunch of green tomatoes and you covered them all with ethylene at the same time? That would happen. They would ripen, and that's how it works in the mass production of fruit and transport from farm to supermarket. The expression of the SRY gene, and again, you're not going to have to memorize this, but you have to follow the story. So by activating the SRY gene through all of these steps, which again, you are not going to have to memorize, are going to form the testes or trigger the male sexual development. What happens if we change these pathways? Well, bad things can happen. So if we change the structures, possibly the cell is not going to do whatever it's supposed to do and could potentially kill the cell. So let's see, a mutation. Again, a mutation could happen, and it will ultimately affect what happens downstream, what I showed you before. So if you have a series of events, that event which triggers that event, which triggers that event. If something happens in the middle, well, then the cellular response doesn't happen. So, for example, herbicides. Got to go out and kill those weeds, right? Some of them will disable the photosystems. And we remember the photosystems from photosynthesis, which is in the last chapter, which is this series of breaking down the water and exciting the electrons and all that other good stuff. And if you mess up that movement of electrons, you inhibit the ATP. No ATP, you could ultimately mess up the ability to synthesize amino acids and or proteins. So messing up one step can definitely mess things up further down the line. Now this one here, these are some just specific examples, but cholera is a bacteria. And if you drink water from whatever, a weirdo stream somewhere in like the middle of a place where you're not really in charge of what's up, it messes up the G protein. Well, what does the G protein do? It is going to, in essence, start this whole cascade. Well, in this case, your body doesn't turn off the release of water and salts. So in essence, you are releasing tons of salt in water, which could cause nasty diarrhea, which could cause dehydration and possibly even death. So, whoa, what happened there? So a lot of this stuff happens in contaminated water supplies. So not good. Viagra, well, that's the opposite. So for example, in males, when the smooth muscles, I don't know what's going on over here. Smooth muscles relax will cause increased blood flow. If you watch enough of these commercials for Cialis and for Viagra, increasing blood flow will help a male get an erection. There you go, I said it. Well, what about programmed cell death, apoptosis? This could also be a cascade. In these cases, the cascade, again, you're not going to have to know this, suicide proteins and what they're going to do. For example, if there is a cell that is possibly damaging neighboring cells, the body's like, got to get rid of it. Programmed cell death. Other examples are the tadpoles, these little guys that before they turn into frogs, they got to lose their tails. And also in humans, when you were in development in your mom, you had skin between your fingers. Now you don't. Other malfunctions that could be linked to later on issues, Parkinson's and Alzheimer's. So those are signal transduction pathways being messed up. As usual, series of videos. Hopefully you watched them. And now let's take a look at the FRQ. There were 4 possible things you could have said for A, so I just asked you for 2. B, a lot of people missed this. You didn't get the question. It says describe the ligand. Don't tell me about it. Tell me what kind of it is according to the diagram, and I'll tell you what that means in a second. Part C, everyone did pretty well on that. And herbicides, I mean, most of you have no idea how that works, but if you would have watched the video, watched the slideshow, the answer was in there. So I was very upset when people didn't get that, because that means I know you didn't read the slides. So this is a classic AP style question. Again, it's simple. This was FRQ a couple years ago, and I've used the picture like this in the past. So you'll need this for B and C. But for part A, pretty simple. I gave you all of the answers, so you have just pick 2 of them. So if you said autocrine, that's where you will release a chemical that will act on that same exact cell. So that was kind of a hard one. It really wasn't a good example. Juxtacrine, they are physically touching. So I was looking for things like plasmodesmata. I was looking for gap junctions. I was looking for materials to flow between cells, things like that. In paracrine, I was looking for very close to each other, but not touching. I was looking for neurotransmitters and nerve cells. That would have been good. And then if you said endocrine, I was looking for long distance. I was looking for hormones in the blood, released from one area, work on another area, things like that. So those are what I was looking for full credit in that question. Now, part B, all you needed to say was this was a protein. And if you wanted to, you could say that the receptor was located in the cell membrane and the ligand does not enter the cell. So if you go back to that picture, notice the ligand, which is this light gray color, does not enter the cell. So that's where many of you, if you lost a point, that's where you lost it. Step C, part C, most people did very well on this. All you had to talk about was reception. And then I would have taken something along the lines of transduction or something along the lines of amplification, which I don't think anybody said. And then if you said cellular response, you got full credit on that. So most people did well on part C. And then, of course, part D, if you would have read the slide, you would have had this. And we could have talked about disabling the photosystems, and once they're disabled, there's no ATP, no Calvin cycle, no glucose, the cell dies. Simple. Or you could have said messed up the synthesis of certain amino acids. You did not even have to be specific. But if you would have read the slides, you would have got that one right. So that's why I do it. So hopefully that was helpful, and I'll see you in the second part of this chapter.